The overall goal of this procedure is to examine the interactions between rare prostate cancer cells and E-selectin expressing endothelial cells using a microslide flow assembly. This is accomplished by first coating a microslide with fibronectin. The second step is to culture human umbilical vein endothelial cells, or HUVECs, in a dynamic flow system on a microslide with a small channel width. Next, the prostate cancer cells are labeled with an anti-prostate-specific membrane antigen, or PSMA, humanized monoclonal antibody, which gets internalized. The final step is to perfuse the anti-PSMA labeled prostate cancer cells over the E-selectin expressing HUVECs. Ultimately, video microscopy is used to show the interactions between prostate cancer cells and the endothelial cells.
The main advantage of this technique over existing methods such as parallel plate flow chamber and other dynamic assemblies is that with this technique you can examine the interactions between rare prostate cancer cells such as circulating tumor cells and cultured endothelial cells in a dynamic flow system. This method can help answer key questions in the field of metastasis, such as how do prostate tumor cells metastasize through the blood vascular system? Do they use the same mechanism as being used by leukocytes during their extravasation? Generally, individuals who are new to this technique will struggle because it involves culturing a monolayer of endothelial cells in a narrow channel under perfusion.
Under the tissue